Well, thanks for Yunfu to give a very comprehensive introduction of the fantastic stereo-seq technology. And I'm happy today to share one of our recent study by using this technology to study Alzheimer's disease. So for some of the audience that are not in the Alzheimer's disease area, so give you a very brief background. So Alzheimer's disease is a neurodegenerative disease associated with progress of memory loss and reduced cognitive function and associated with a number of different behavior changes. It accounts for about 60 to 70% of the dementia. And at the cellular level, there's two well-known neuropathological hallmarks of AD. One is the accumulation of the amyloid beta plaques outside of the neuronal cells. And another one is the intracellular accumulation of the neurofibrillary tangle, or in short, NFT, which is composed of the hypophosphorylated tau proteins. So over the past couple of years, with the advantage of the single-cell or single-nucleus RNA, as well as the spatial transcriptomics technology, there's several remarkable papers have been published, delineated the very diverse molecular landscape of the brain cells under the normal aging or Alzheimer's disease patients. So most of these studies actually lack the capability to capture the spatial transcriptomic information and single-cell or single-nucleus resolution. So equipped with the stereo-seq technology, so we actually carried out a kind of pilot study to characterize the spatial transcriptomic analysis and the single-nucleus resolution between the AD cases and controls. So this is the basically study flow of this project. So we obtained brain samples from 12 subjects, including 6 AD cases and 6 controls. These are highly matched. I will show you some of the characteristics in the next slides. So we focused on the one specific brain area, which is the Brodmann area 10. This is a region that's highly vulnerable during the aging and Alzheimer's disease. So and we applied the stereo-seq into these to study the spatial transcriptomics in this area. And as Yongfu mentioned, this stereo-seq, you can have the different ways to group your the so-called bins. Then you can study the transcription profile at a different scale. So we actually did on this take advantage of this unique feature and studied this on the different scale. And well, of course, we applied a series of sophisticated bioinformatic analysis to study the expression profile changes. So this is a table showing the kind of detailed information about the 12 samples. So like I mentioned, there's 6 AD cases and 6 controls. The AD cases and controls are closely matched with the sex, ethnicity, and age. So they are all males from the non-Hispanic or white, and the age range from the 70 to 90. So within this AD group, we actually further separated into them 2 subgroups, which is a moderate AD or severe AD groups. So these are actually based on the immunohistochemistry staining of the A-beta plaque. There's 2 slides, 2 images showing the example. So the red dots represent those AD plaques staining positive. So like I mentioned, we started this analysis with and kind of like a different scale. So first we want to look at a kind of a larger scale to see whether we see any transcriptomic changes. So we focused on the so-called cortical laminae, which is, this is give you some background. So the human cerebral cortex is normally morphologically organized into 6 layers, or we call them laminin. So each layer was different cellular composition and intra or interlayer connectivity. This is basically a schematic illustration of the 6 layers ranging from the layer 1 to layer 6. That's start from the surface of the brain and moving inwards. And after the 6th layer is basically the white matter. So back into the year 2021, there's a paper published by Maynard, etc., and they used the 10x Vision platform to do the spatial transcriptomics in a healthy human brain sample. So they actually aligned the transcriptomic data with the Jensen H and E staining to identify to delineate the 6 layers in their transcriptomic data and identify some specific markers for each layer. So the first step we want to do is kind of to compare whether we can identify similar structures in the brains in our sample. So to do this, we converted the raw data into the 55 micron so-called pseudo spots, basically a beam. So each one, we choose this resolution because we want to have this comparable with the Maynard paper. So this one, each one of the pseudo spots basically similar to one spot on the 10x Vision platform. And then we apply the newly developed unsupervised clustering algorithm, which not only clusters cells based on the gene expression similarity, but also can take into consider their spatial proximity. So indeed, this is showing some of the results. These are the 4 controls, different controls. There's 2 moderate AD sample. So indeed, we identified 6 layers as well as the white matter based on the transcriptomics profile. And the layer 2 and the layer 3 are pretty similar, so we actually grouped them into one layer here. And the next, we basically perform the differential gene expression analysis between different layers to identify specific markers for each layer. And this heat map here showing some of the marker genes we identified, and some of the marker genes we highlighted here, which actually matches with the marker genes identified by Maynard. In addition to those, prior known marker genes, we also identified some novel marker genes for those layers too. Next, we wanted to see, interestingly, we see that in the severe AD group, the transcriptome-derived laminar structure is actually largely disrupted. If you can see here's 2 images from the severe to severe AD sample. So you can see, although the algorithm still identified the different layers, but you can barely see any clear separation of the 6 layers. And then we summarized the different proportion of the layer in the 3 groups, that's in the control and the moderate AD and severe AD. So you can see compared to the control, the moderate AD, the different proportion is quite similar, but in the severe AD groups, there's the laminar structure are largely disrupted. So we think this disruption of this transcriptome-derived laminar structure is likely related to the extensive neuronal degradation and the significant reactivation of astrocyte that is a known feature for the later stage of AD. And this is also kind of, hypothesis is supported by the observation of the upregulation of the GFAP and NE81, which is the traditional markers for the reactivated astrocyte. Of course, we know that the communication between different layers in the cortex is important for the information processing and the supported the cognitive function. So we want to study the layer-to-layer communication network between the different between the case group and the AD groups. So to do this, we constructed a layer-to-layer communication network based on the database of interaction among the ligands and the receptor. So this plot here shows the total number of interactions we identified between the different groups. So you can see with the progression of the AD, there's a decrease, generally a decrease of the total number of the ligand and receptor interaction across different layers. And we think this is possibly reflecting the collapse of the neural connection network due to the neuron degeneration in the AD group. This two figures showing the more detailed kind of like pairwise comparison between the moderate AD and the severe AD versus the control and across different layers. So, so the y-axis shows the outgoing signal, basically the ligands, and the axis is showing the incoming, basically the receptor signal, and the different colors represent the increase, which is red, or decrease, that is shown as a blue, of the number of the interactions we identified between different layers. So I want to highlight that we see during this kind of area that which is the outgoing from the layer 4 and 5 to the layer 2, all the way through layer 6, showing the great decrease in the severe AD group. And we know that the layer 2 to 5 is critical for information collection and processing. So we think this reduced interaction between these layers possibly reflects the breakdown of the vertical network, and which actually contributes to the cognitive impairment in AD. Next, we actually switch to a different question. So we know that during the normal aging as well as the AD, the brain cells normally become increasingly vulnerable to the oxidative stress or inflammation, and this actually contributes to the accumulation of A beta plaque and the neurofibrillary tangle, which actually in turn intensifies the neural stress and causing the neurodegradation. So we want to ask, how the brain cells and actually responded to the different stress in a spatial and a cell type specific manner. To do this, of course, first we have to identify where those brain regions under high stress is located. From prior knowledge, we know that excitatory neurons are highly vulnerable to stress in both normal aging as well and in AD. And there's known positive correlation between the cell degradation and the increased mitochondrial gene proportion. So we presume that excitatory neuron with high mitochondrial gene proportion would reasonably corresponds to the brain area under significant stress. To do this, this is basically a simplified workflow. So we did cell segmentation at the single cell or single nucleus resolution. Then we identified, after cell clustering, we can identify those excitatory neurons, and we can calculate the mitochondrial gene proportion. Then we can basically pinpoint those areas, brain areas under high stress. Then we can, using the so-called concentric center analysis to categorize cells into different categories based on their space proximity to those stressed area. Then we can compare the gene expression profile between these cells that are close to or distant to those stressed brain areas. So, like I said, the first step is to identify those areas, brain areas under high stress. So to do this, we regrouped the gene expression data into these 25 micron pseudospots. This is roughly the single cell resolution. The reason we do this is because to calculate the mitochondria gene expression, you have to cover the cell cytoplasm. So to do this, so we used this about a rough single cell resolution, so we can cover both the nuclei as well as the cytoplasm. Then we applied the cell segmentation algorithm to identify those different brain cells. These are shown here as a UMAP. There's different cell types, such as the astrocyte, endothelial cell, excitatory neuron, and the inhibitory neuron. Then we can calculate the mitochondrial gene ratio in each cell. Basically, that's the number of mitochondrial genes over the total number of detected genes. And because there's no consensus of how much of the percentage of the mitochondrial gene proportion is kind of like too much. So based on the data distribution of our data, so we use the relatively arbitrary cutoff line, which is 15%. So to define those, these are considered a high mitochondrial gene proportion. So with that, so basically we can identify those brain areas that contain the excitatory neuron with over 15% mitochondrial gene ratio, and we consider these as the brain areas under high stress. So this one shows the different 3 pie charts into the, I apologize that I missed some of the footnotes here. So this one is the control group. This one in the middle is a moderate AD group. This one is a severe AD group. So, and the blue region represents the proportion of with a extra excitatory neuron with normal stress or kind of a lower stress, and the brown area represents the proportion of the excitatory neuron under high stress. So we can see here, compared to the control groups, that the moderate group have a very modest increase of the excitatory neuron under high stress. But if you look at the severe AD groups, there's a significant increase of the area under high stress. So we think this, with this actually support our assumption that this excitatory neuron with over 15% mitochondrial gene proportion reasonably represents the brain area under high stress. Next, we want to bring this, all the way to the single nucleus resolution. So so based on the nuclear staining image, which is actually provided along with the stereo-seq data by this technology. So we applied a deep learning model to do the nuclear segmentation. This one shows you one of the examples showing the cell segmentation on the nuclear staining image. So after QC, we analyzed about 400,000 nuclei. Each nucleus contained about 350 genes detected. And we performed a clustering analysis to identify the nuclear subgroups and annotated these clusters into different, basically, cell type or nucleus types, as shown here. The next step, like I mentioned, we did this concentric center analysis. Basically, we classify these nuclei into 3 different categories based on their proximity to those highly stressed brain area. So in the level 1, basically, they're in the center. So they are less than 250 micron to the stressed brain area. The level 2 would be the area between 250 to 750 micron, and over 750 to 1250 micron would be considered level 3. And here is showing one of the example showing in one moderate AD sample. And the red dots represents the nucleus at a different level from level 1, 2, and 3, so the locations. So next we actually we wanted to compare the transcription profile between these level 1 and the level 3 cell. The reason we can focus on these two is that actually we found the expression profile between the level 1 and level 2 are very similar. So basically here we only focus on level 1 versus level 3 to represent those cells are close to those stressed neuron versus those are relatively distant. So the first analysis we did is the gene expression, differential gene expression analysis. And here shows the, if you look at the control here in the top panel, shows the level 1 versus level 3. And this is a different color represents the relative gene expression level. So with the red color represents the higher expression. So we can see here in the control group, the differentially expressed genes are largely nucleus type specific, so meaning that the differential genes are largely different between different nucleus types. I want to highlight one of the group of genes here is the higher expression of the neuroprotective gene, such as the VGF or RMO4 in the level 1 versus level 3 excitatory neuron in the control group. So this roughly reflects that the cells close to those stressed neurons are having higher expression of these neuroprotective genes, possibly to mitigating the stress of this neuron and trying to prolong the neuron survival. However, if we look at the severe AD group, we see a whole lot more that's differentially expressed genes that are common in different nucleus types. For example, if we're highlighted here, there are some of the heat shock protein genes, like HSPB1, HSPA1A. They are the increased expression of these genes are not only observed in the excitatory neuron, but also in the astrocyte and the inhibitory neuron, as well as oligodendrocyte in those areas, in the areas that are close to this stressed neuron. So this is actually consistent with some of prior knowledge that we know in the early stage of AD, most of the gene expression changes are cell type specific, whereas in the later stage, there's more common expression changes across different cell types. We know the intercellular interaction is very important to support the proper cognitive function. So we started a intercellular interaction analysis among those cells that are proximal to the stressed neuron in controls, as well as those nucleus in the level 3 that into different groups. So in the left side figure, we can show this is the number of inferred interactions in different groups in the level 1 and the level 3. So we can see in the control groups, we see a large number of intercellular interaction observed in controls. This is possibly reflect as a protective mechanism against the degradation of the stressed neuron. However, if you look at even in the control group, if you look at the cell nucleus that are distal to this stressed neuron, we didn't see there's a lot of interaction. In contrast, in the Alzheimer's disease group, in both moderate and as well as the severe AD group, their intercellular interaction is largely disrupted. Even in the level 1, you see there's a significant decrease of the intercellular interaction between those different nucleus types. This right panel basically shows the example of the level within level 1 in the control group of the severe AD group, the detailed interaction between different nucleus types. Each of the dots represents each one's nucleus type, and the connection between those represent the potential ligand and receptor interactions, and the thickness represents the number of the interactions between those different cell nucleus types. So we can clearly see the interaction, the intercellular interaction in the severe AD group is disrupted. We can actually take a closer look of some specific ligand and receptor interactions. Here is an example of the APP and CD74 interaction, which is actually known interaction, that this interaction, the APP released by the neuron will interact with the CD74 on the microglial cells, and it actually functions as to inhibit the A-beta production. So we can see there's in these kind of plots, the different section of the ring represent different nucleus type, and the connection between those different nucleus types represents the interaction. So we can see, in the control group, in the cells, close to the stressed neuron, there's a lot of interaction between the neuron and the microglial cells. However, if we look at the level 1 nucleus in the severe AD group, we barely see any interaction. So this shows this specific ligand and receptor interaction are significantly diminished in the severe AD group, and the loss of the cell-cell interaction in cells proximal to these highly stressed neurons in AD possibly contribute to the formation of A-beta plaque and exacerbated neuron degeneration. In addition to these concentric circle analysis, we also performed some gene co-expression network analysis, because we know that genes normally do not function, in isolation. They actually function. The genes kind of, collaborate with each other in those networks. So so we first performed the so-called weighted gene co-expression network analysis, or in short, WGCNA, to construct the gene co-expression networks, or in other words, also called modules. So each of these genes in these modules normally their expression profiles are highly intertwined, normally reflects a common functional activity. So we applied this method to the 2,000 most variable genes for each nucleus type across different sample, and we identified one module in the excitatory neuron and two modules in inhibitory neuron. Here shows one of the example of the modules we identified in the excitatory neuron, and it also shows some of the so-called module genes, the so-called hub genes, and we also highlighted some hub genes that are known to be related with AD. So to understand the possible function of these modules, we performed the so-called gene ontology enrichment analysis in the 3 modules. It turns out all the 3 modules were primarily associated with the neuroprotective process and the inhibition of the formation of AD pathological hallmarks. There's some of the biological process, such as the negative regulation of amyloid beta formation, negative regulation of protein phosphorylation, and positive regulation of dephosphorylation. These are all point out to these 3 modules all shows the neuroprotective effects. We also used these so-called eigen genes to see whether these 3 modules are actually associated with AD progression. It turns out all these 3 modules are negatively correlated with AD, and implying again that they are protective to the neuron degeneration, and possibly just have the capacity to diminish they are implying the negative association implying a diminished capacity of A-beta clearance and the tau dephosphorylation during the AD progression. And the next step we did is to trying to identify some transcription factors that are enriched in these gene co-expression module. And so we applied this algorithm called a motif scan to identify the different transcription factors enriched in each module. I want to highlight one specific transcription factor called ZNF 460. This transcription factor actually shows the highest level enrichment across all the 3 modules. However, its function in the brain and this time is unclear. So we're certainly wants to follow up on this transcription factor to see whether this may represent a potential treatment target for the Alzheimer's disease. In summary, we used the Stereo-seq technology to generate a first comprehensive high-resolution spatial transcriptome atlas in the prefrontal cortex for the AD and the healthy controls, which represents the normal agent. We identified significant changes in the cellular composition and the interlayer communication that are associated with AD. We also reviewed the gene expression and cell-cell interaction in response to stress during the normal aging and the differential AD stage. And we discovered a novel transcription factor that may function as a master regulator for those gene networks that are associated with AD. So this is, I want to thank all those members who contributed to this work. There's a talented group of graduate students and scholars and the Tulane University, the Center for Biomedical Informatics and Biomedical Informatics and Genomics, Dr. Harry and Dr. Swallow and the University of Kansas Medical Center provided the precious brain samples. And there's investigators from China as well as the University of Michigan to help with the analysis of the data. In the last but certainly not least, I want to thank the general support from the STOMICS program by the Complete Genomics. Thank you.